Thank you very much. It's a pleasure to be here. My name is Lukas, and I'm one of the founders and the CEO of Hemotune. And as you heard, we are disrupting blood purification. But let me start maybe with drugs before, because drugs is how we are currently treating disease typically. And how do we do that? We are actually adding something to the body that is in many cases toxic. So we know that it's toxic, but actually we dose it in the right way. And then these things, these drugs, they interact on biological cascades. They choose one pathway, and by interrupting or changing this pathway, they are adding value or treating disease. But what we can't do with drugs is we can't act on multiple pathways in parallel, and as we're inside the body, we have many of these off-target effects or side effects. However, there's also another way of treating disease, and it's through removing the bad compounds from the blood. And this was used for a very long time. Bloodletting was already used in Egypt almost 3,000 years ago, and for thousands of years, this was actually the magic bullet of medicine. You would bloodlet for everything, and doctors would believe that this is just like the cure for everything. About 200 years, this stopped. We realized that probably just taking out blood doesn't help in any condition. And about 80 years ago, we started to reuse blood purification mainly for dialysis, for kidney replacement therapy. And we have come up with dialysis machines, and in the last 40 years, there was actually very little innovation. So we came up with the dialysis machines to purify blood for the kidney replacement, but there was really little innovation in it. And that's actually a pity, because blood is very unique. It's the transport system of our body. It's full of different compounds, cells, thousands of proteins, thousands of metabolites, millions, billions of different antibodies, and it's the way how everything is distributed in the blood. So it would also give us a huge potential to access disease and to interact by removing stuff from the blood. And I think as we're also understanding diseases much better, it offers a huge opportunity to access blood, because now, in these times, we are understanding diseases better because we can test better, we can do metabolomics, we can understand also networks much better. With AI tools, we can analyze large data sets, and we can find out what leads to what. And with this development, what becomes clear is that many complex diseases, they are not monocausal. There's not one thing that leads to the disease progression without anything else involved. And if you look at this network, this is actually a simplified network of aging. It's also clear that probably by just taking out one arrow, we will not solve aging. Nobody would think that if you are just like cutting one of these arrows that the problem will be solved. So I strongly believe that we need new treatment methods to actually deal with this complexity and being able to really steer complex diseases. And at Hemotune, we are working on two main things for blood purification. It's the selectivity. Why is selectivity so important? Because there's so many things in the blood that are actually essential. So there's a lot of good things in the blood that we don't want to touch. And at the same time, to treat complex diseases, we probably need multi-target capabilities. So we need to be able not to only take out one thing, but a couple of things to really have synergistic effects and improve these disease progressions. That's why we founded Hemotune. We're a Swiss-based company. We're now 25 people, and we have developed the blood purification machine that you can see here also on the picture. And as it's such a new field, we also worked a lot with regulatory authorities, because we are not a drug, we are a medical device, but we are really having like biotech-like intervention strategies by removing stuff instead of adding stuff. We're also fully certified under ISO 13485, so the medical device regulations, and we have just completed also our preclinical studies for our first indication, which is in sepsis-induced immunosuppression. And I'm going to talk about two programs, one in sepsis and another one which we have successfully completed feasibility, which is in an enabling technology. It's enabling gene therapy. But let me start with a brief video about our technology.
Sepsis happens when an infection in or on the body triggers a life-threatening, misguided immune response. Because 1 in 5 of all global deaths are sepsis-related, and almost 50 million people suffer from sepsis every year, it was declared a global health priority by the World Health Organization. To treat sepsis and other bloodborne diseases, Hemotune has developed a disruptive blood purification platform for the use in intensive care units. In a device similar to a dialysis machine, nano-engineered sorbent beads remove disease-related compounds. The beads contain a magnetic core, a non-sticky polymer coating, and affinity binders that attach to the desired targets. By combining different beads, Hemotune is able to perform precision medicine multi-target interventions, which would not be possible by traditional blood purification or through the use of drugs. With a size 300 times smaller than a red blood cell, the beads selectively attach only to the target compounds. While passing through a magnetic filter, beads and bound targets are highly efficiently removed, and the purified blood is returned to the patient without containing any beads. Hemotune's goal is to restore adequate immune function in sepsis and improve patient outcomes.
So as you can see, it's quite similar to a dialysis machine. We pump the blood outside of the body, but instead of having a cartridge or a filter, we really add these magnetic beads. They very selectively bind to the disease-related compounds, and they are removed magnetically. And as it's a platform, we can basically fine-tune the surface of these magnetic particles to target different compounds, while we can reuse the device and the disposable across different indications. And you heard sepsis is the first lead indication, and it's a huge problem affecting a lot of patients, 11 million deaths. But it's not only a problem for these affected patients, it's also a huge problem for the healthcare systems. We heard yesterday the talk about how much we spend not preventing diseases. Actually, sepsis is quite similar. In sepsis, we spend 0.33% of global GDP on just treating these patients in the intensive care units. And we have looked at how sepsis was seen for a long time, and it was always seen as a hyperinflammation, an infection that leads to an overreacting immune system, and the strategy was always to dampen this immune system. However, this did not really lead in improved survival rates. It just shifted mortality from early mortality after 1, 2, 3 days on the intensive care unit to after 2-3 weeks on the intensive care unit. And this is, of course, a huge problem. We are talking about health span, right? Like having a healthy life. And these 2-3 weeks in the ICU is absolutely the opposite. You're in a coma, and you're just like, kept alive by those machines. So we have teamed up with immunologists and key opinion leaders in sepsis to develop an intervention strategy based on the most or the newest understanding of how sepsis evolves over time. And we have from left to right the time axis, the disease progression, and from bottom to top the immune status. And sepsis starts with a hyperinflammation, so an activated immune system, but only few patients actually die in this phase. They all undergo a counterbalancing immunosuppression, and there, the interesting thing is that 1/3, they regain an immune balance, and they actually have much better outcomes. They have better chances to recover. However, the 2/3 that stay immunocompromised, they stay in the intensive care unit for the longest time, have the highest mortality, and also incur the highest healthcare costs. So we are selecting those patients based on an immunobiomarker, and then we are starting to treat to remove multiple factors that lead to this immunosuppression, to restore their immune function, shorten the length of stay in the ICU, and improve their survival rates. It's also a very big market, many patients affected, and also because it's such a long time on the ICU. So that's the first program, but we have also another program that we're working on, and that's more an enabling technology. And you all have probably heard about gene therapies. What's the idea? We are implementing or changing a gene in the body that is faulty, that doesn't produce the protein, or has a problem. And it's a very powerful tool, and what we need is something to bring in the genes into the cells. And we do that with vectors. It's just the delivery vehicles. And the most commonly used vectors are actually viruses. AAV viruses, they are naturally occurring viruses. They are everywhere. They infect humans, so they are introducing genes into the human cells, but they don't make sick. And because they deliver genes without making sick, they are the favorite tools in gene therapy. Actually, they are 82% of all gene therapies under development are AAV-based. And as you all know, gene therapy is an emerging field, so it's a lot of assets that are currently in the pipeline, and only few on the market. The ones that are on the market can cost up to $3.5 million per treatment, because they are so powerful. And also here on the market that we have 86% of AAV-based gene therapies. There is a big problem in these AAV vectors, and it comes with the biggest benefit. Because these vectors are everywhere, our immune system knows them, and our immune system has antibodies against them. And if you have antibodies against them, the gene delivery is not efficient. So there's two things. First, the antibodies, they block the vectors from delivering the genes into the cells, but on the second side, the complexes, antibodies and vectors, they are recognized by the immune system, and they lead to an immune reaction that can have very severe side effects, and there were also, unfortunately, some death from these side effects. So what do you do currently? You are screening the patients, you're measuring the antibody levels, and if this antibody titer is too high, we can't treat those patients. And as we have huge numbers of these AAV antibodies in the population, 30 to 70%, of course, we don't want to exclude a majority of people from these potentially life-saving therapies. So we have done two things. We have looked at, can we remove those antibodies in the first place? And then when we remove the antibodies, does the gene therapy work better? And we can really very nicely reduce the antibodies from very high titers. So you always have a cutoff to which titer you can still use the gene therapy. In this case, we used Zolgensma, an approved gene therapy. And what you can see is we can reduce all the titers from non-eligible patients to eligible patients. And also, the second slide, it's another product under development. We can also here reduce extremely high titers to enable then potentially gene therapy dosing. And the idea is really that in the future, this will become the standard of care for gene therapy. You measure the patients, and if it's too high, you would hook them up to our machine, you reduce the antibodies, you can dose the gene therapy, the antibodies are reformed in the immune system. We also only take out the antibodies against these capsids, against these vectors, not like destroying the whole immune system. And then we could also allow redosing. So it could allow us to have a faster clinical trial enrollment, expand the access of gene therapies for the patients, lowering the doses, because now a lot of the capsids, they don't end up in the cells because they are intercepted. And we could also allow a better efficacy, safety, and potentially redosing. So with that, we believe we can really have a huge impact on gene therapy in the future. So now we are entering the clinical phase with our sepsis program in a study with up to 204 patients, while we are also expanding the platform to these additional indications. We have a very interdisciplinary team. It's a complex product, software, mechanics, electronics on the one hand side, the disposable, and also all these nanoparticle developments. So I'm very glad to have a great team behind that develops these different aspects of the technology. So let me finish with just like this last slide. I think we really have a better understanding of complex diseases, and this understanding will even grow in the next years. And AI will probably help us quite a bit in understanding these networks. And many of these complex diseases will not be monocausal, so we need multi-target strategies. So the next point is really removing is probably better than adding something. By adding something to the body, you will induce also side effects. You will induce immune recognition. And by these complexes that for example, form with antibodies, you're creating new problems. So we believe removing might be much more elegant than adding something to the body. And then the third thing is really the selectivity. There's a lot of very good stuff around, and we need to be able to pick only what we want to pick, but we need to also be able to remove multiple things at the same time. So with that, I want to finish my talk. Thank you very much, and I'm very happy to answer any questions.